These are all going to be 3-minute talks. We're going to have a time for one quick question if everyone sticks to the 3 minutes in between. So we will try to run the microphones up and down the aisle real quick. Okay. We'll go ahead and get started. So first we have Nikko from LSU.
Yeah, thank you. So there was a lot of interest about these events, precipitation events. This is one example. We had Hurricane Harvey in August 2017. Extreme precipitation that causes flushing of suspended sediments, suspended material to the coastal ocean. So in response to that, we went on 2 field trips, 1 month and 2 months after the hurricane. And the idea is to look at the biogeochemical response. As you can see, one of the big issues is that estuaries are optically complex and standard ocean calculators don't work, the algorithms. So we developed empirical relationships and semi-analytical algorithms tuned for estuaries, and outcome was absorption spectrum, backscattering, and using set of these relationships to derive CDOM and backscattering, SPM and POC, and also DOC. So we had a set of imagery related to DOC, as you can see before and after the hurricane, and backscattering. Using previous relations, we derived DOC and POC. We also had hydrodynamic data, currents and tides at the bay entrance, and we actually calculated over 10 days the total fluxes out of the bay into the coastal shelf. We get about 25 million kilograms of TOC, 314 million kilograms of SPM that were rapidly exported from the bay to the shelf waters. And finally, we also looked at phytoplankton response using inversion algorithms, and we found that under freshwater conditions, we had larger phytoplankton species, and after 2 months under more saline conditions, we have small-sized phytoplankton, and verified this with pigment maps using another inversion algorithm. Overall, we got a high response, biogeochemical response to the floodwater event.
Any quick question while the next speaker comes up? We're good? Okay, we'll go ahead with Kevin Kroger from USGS is going to give us a little review of the Wetlands Lateral Flux Workshop.
Okay, so I'm giving a report on an OCB supported activity titled Lateral Carbon Flux in Tidal Wetlands, filling a key knowledge gap through a methods intercomparison and data synthesis. And last August, we had a workshop with about 29 individuals who have expertise in related topics. And the basic idea is that the coastal zone is increasingly recognized as an important but difficult to quantify component of the carbon cycle. Wetland lateral fluxes are poorly known, and so what we mean by lateral fluxes are aquatic tidal exchanges of dissolved organic carbon, inorganic carbon, or particulate carbon. So you can see in this diagram here from a Najar et al. Paper recently that tidal wetland fluxes to estuaries are a significant part of the estuarine carbon budget. So the objectives were to review and improve methods and to estimate annual rates of carbon exchange across the contiguous United States. And we began with the following 2 hypotheses: that lateral flux is related to the rate of carbon cycling within the wetland. So that would be something like GPP or respiration. And lateral flux is related to the rate of aquatic exchange between the wetland and the estuary. So these are based on logic. There's not enough data to test these yet, and we're going to try to test those hypotheses. So we addressed and suggested approaches to the following complications that interfere with measurements of annual net rates. First is that the carbon budget doesn't close. Classical methods to measure attempt to measure a continuously varying flux based on low-frequency measurements. And you can see from this brief trace here over a couple of weeks that there's a lot of temporal variability in the flux. And so you need high-frequency continuous data. The contributing area to the flux from a wetland, indicated in this case by this heavy black line, is often not known, so that a per unit area flux is difficult to quantify. We arrived at two approaches to estimate the lateral flux, the approach going forward is to test relationships to measures of carbon supply or cycling rate and intensity of aquatic exchange. And based on those relationships, model and map the lateral flux based on mapped GPP, respiration, and tidal exchange. Or if that approach is not effective, then we will essentially calculate a budget and try to close the budget based on difference. And progress has occurred along three main fronts. First, we have new and compiled field data. This is the most difficult part of the effort, to get high-quality annual flux rates from a number of different kinds of sites around the country. We're moving forward on that. We have comprehensive information developing on marsh productivity based on a remote sensing model that's being led by Rusty Fegan and Inka Forbrish. And we have comprehensive data now on intensity of tidal exchange based on an effort by James Holmquist et al., in which they're quantifying marsh platform elevation within the tide frame. And so those fluxes will then all be related to amount of wetland coverage in each of the main estuaries in the United States to produce a map of flux. Thank you.
Okay, any quick question for Kevin? If not, we'll move on to Julia Moriarty. Great. She will, from, oops, sorry. How do I cancel it? She's going to be speaking on redistribution of particulate matter following marsh lateral erosion in a back barrier estuary.
Okay, thanks, Margie. So for those of you I haven't met, I'm a postdoc at the USGS with a background in sediment transport. And so for my postdoc, I've been looking at one small piece of the puzzle that Kevin talked about and Ray talked about earlier. And it's the fact that marshes store a lot of carbon, but they also erode. And so the question becomes, where does this material go, both the sediment from and the particulate organic carbon? And we're interested in this from a carbon point of view and then also from a geomorphology point of view. How will the shape and the extent of marshes change in the future? And so in this process is complicated because if you look at these realistic systems, there's a lot of variability in space and also, as Kevin mentioned, in time. And so we've been developing a numerical modeling approach that can help. We've been better adapting a numerical modeling approach that can be used in these coastal vegetated systems. So in particular, I'm working with Neil Gandrew, and his group has been incorporating vegetation hydrodynamic interactions into coast, which includes ROMS in it. ROMS is the regional scale hydrodynamic model that a few of the speakers used yesterday. And so for this, and the reason we chose coast and ROMS in particular is because it's an open source, widely used model by the community. And then it has the biogeochemical and sediment transport processes in it, and we've been adding vegetation hydrodynamic interactions, as well as now marsh edge erosion. And that's just this process you see here where waves are eroding the edge of the marsh. And so I'm not going to go too much into the details of the results. If you are interested, come talk to me. But in short, what we're seeing is when we implement the model for some of these back barrier estuaries, we're getting variability in sediment fluxes between areas near these inlets compared to other areas of the estuary. So where you have energetic waves and tidal currents moving, you have higher export, both a higher erosion and then higher export of material from the marshes. In contrast, then as this material is distributed through the estuary, you're getting higher fluxes into the marshes where you're getting higher sediment supply, so here as opposed to here. Thank you.
Great. Any?
Margie?
Yes.
I have a quick question.
Yes.
In your model, does the topography change? Does it evolve when it erodes?
So it depends how you set it up. So as I'm currently using it, no. But it's set up to do that, and Taran Kalra at USGS, who was one of the collaborators that was on my slide, he's using it to do some geomorphic change, idealized studies currently.
Cool.
So it's still in development a bit.
Okay. Anything else? Okay, great. So next we have Jenny Thomas from WHOI talking about the impacts of estrogen dynamics on CO2 air-sea exchange.
Yeah, hi everybody. I'm a postdoc here working with Malcolm Scully, and the reason we're interested in this subject is, as you probably figured out by now, that estuaries are proposed to play an important role in the global carbon budget, but there's still a lot of uncertainties about spatial and temporal variability of CO2 air-sea fluxes. And so we're using a model to look at some of this. It's a model of the Chesapeake Bay using the regional ocean modeling system that Julia just mentioned. And this model reasonably accurately reproduces observations of oxygen, which Malcolm showed yesterday. And here I'm just showing a plan view of the annually averaged O2 on the far left and CO2 in the middle, air-sea fluxes from the model output. And what we can see is there's spatial variability. So the blues are negative, those are fluxes outward. And so for the CO2, you can see that in the upper bay, there's a big flux out, and that decreases as you go down the bay. And one of the reasons, physical reasons, is because stratification is limiting CO2 outgassing in the lower bay. In the right panel, I'm just showing the sum of the oxygen and carbon dioxide fluxes, because in my poster I was looking at where these two things differ from each other and why those might be. The annually integrated flux of CO2 from this model is CO2 flux outward to the atmosphere, but there's also temporal variability, and so here I have the area, or the spatially and daily integrated air-sea fluxes of oxygen in blue, CO2 in red, and net ecosystem metabolism in yellow. And so sometimes the direction of flux is in agreement with the net ecosystem metabolism. So for example, on day 31 on the far left, it's net autotrophic, and we have CO2 fluxing into the water like you might expect. But there's also numerous times in the model where the CO2 fluxes are smaller than or even opposite sign than we'd predict with net ecosystem metabolism. So for example, in the later part of the year, we still have really big O2 fluxes, but the CO2 fluxes have gotten really small. And so we're interested in understanding if that could be important for storing carbon in the bay in that period of the year. Since posters are already over, if you're interested in talking to me more about this, I'd be happy to speak with you. Thank you.
Talking on characterizing the spatio-temporal PCO2 dynamics associated with water masses in the river-dominated East China Sea.
Okay, thank you. Okay, good afternoon, everybody. I'm Showen Tao. I'm a first-year master student at the National Taiwan University, and today I will share some of the works that our lab have been doing in the past 10 years. So our lab is focusing on the East China Sea, the PCO2 air sea flux in the East China Sea. The East China Sea is an important area because it's largely influenced by the Changjiang, which is one of the world's largest river and supports a lot of high nutrients and high primary production in the area. So we want to look at the spatial and temporal patterns of CO2 flux in a seasonal pattern. So I will just briefly show the results. This is a conceptual pattern throughout a 4 season. So in the spring, where we can show, we can show that the Changjiang River starts to expand, and then this is a strong carbon sink area, And then, but in the outer shelf, which is more dominated by the Kuroshio, we separate the Kuroshio water and the Changjiang water by salinity difference. The salinity under 31 is Changjiang water. So we can see that in the spring, it's more of a strong carbon sink in the river plume area, but because temperature is low, the PCO2 is low, so the outer shelf is more of also a PCO2 sink. But in summer, the temperature starts to rise, and then the PCO2 starts to rise, so the outer shelf becomes a weak carbon source. But at the same time, the inner shelf, which is the river plume area, is still a strong carbon sink because of the biological uptake. And interestingly, this period of strong stratification, so we can see a high DIC under the river plume area, this area. So in the autumn, when the stratification starts to break, we can see a high DIC start to mix up. So in autumn is the area which has the largest carbon source. But when autumn turns into winter, the temperature starts to decrease. So the PCO2 in the outer shelf starts to decrease again. So in the winter, the whole area starts to become a strong carbon sink. So we can see in a simple framework, a carbon sink and weak carbon source, and then back into a strong carbon sink in winter. So by using a 2N member model, a simple model, we can separate the Kuroshio water with the Changjiang water. And then we can see that here it's the delta PCO2, the delta PCO2 in the Changjiang water. Here we just want to show that it's highly dominated by biological uptake. And this is part of my research, which I showed earlier in the poster session. I try to, because Kuroshio water is really dominating a large N member of the mixing in the East China Sea, I want to look at how the DIC is controlled by, like what processes actually control the DIC in a seasonal pattern. And here I actually show that the salinity difference, which is caused by evaporation and precipitation, is also playing an important role in changing the DIC in a seasonal change. So maybe salinity change is also playing a large role other than temperature and biological uptake in the East China Sea. So I know there are among you a lot of experts on carbon processes and carbon modeling, so I'd be really grateful if you would come talk to me, maybe after giving me some advice. We still have a lot of details that we need to work more on, so thank you very much.
Okay. Next we have Annie. Next we have Annie from University of South Carolina, talking to us on temporal changes in nitrous oxide sources and sea air fluxes in southern Bengal.
So this project is in a collaboration with Sarah Fassett at the University of Cape Town. And the rationale for looking at N2O, it's a greenhouse gas, it destroys the ozone. And although we know that the open ocean accounts for 30% of the global emission, the coastal area are still poorly quantified, especially the southern Bengal. So we sampled the station along this transect here of the South Bengal, and during three times, December 2016, February and August 2017. And the three main questions I wanted to answer are what are the sources of water column N2O, what are the temporal variability in N2O distribution in the water column, and also N2O emission. And of course, we wonder what is the sea air flux of N2O significant globally or for the region. So there is different ways to produce N2O. You can produce it during oxic nitrification by either hydroxylamine oxidation or nitrifier denitrification. You can also produce N2O during denitrification under anoxic condition. But you can only consume N2O during denitrification under anoxic condition. So we measure N2O concentration, but also the stable isotope and isotopomer to be able to distinguish between those different processes. So quickly, some results. I don't have the time to go in detail into the isotopes and isotopomer, so Please come see me if you have more questions about that. But what we found is that there was temporal sources and temporal variation in the N2O sources. For instance, we found that N2O was mostly coming from nitrification, denitrification in the sediment in August and February 2017 versus water column nitrification in December 2016. And here I included some plots showing a delta N2O, which is the concentration of N2O measure minus the N2O concentration at equilibrium. And you can see that it's highly variable. This is December 2016, February and August 2017. So, and we can also see that the IS emission, where it's the concentration are IS at the surface, are the IS in February and 2017. And we calculated fluxes of 0 to 9 micromole per meter square per day, which is about 2,000 of 2 orders of magnitude less than what we observe off the Peru coast, but in the same order of magnitude than what we observe in the northern Benguela. So if you want to learn more about N2O in the southern Benguela, please come see me.
Question, or George, are you here? Yeah, coming down. Okay. Excellent. So next we have George Xu from LSU talking about understanding and quantifying carbon export to global oceans through deltaic systems.
Yeah. So the best thing to be a modeler is that we don't have to worry about seasick. So like, for example, the paper I published here, when I first finished the draft, I never been to the Gulf at all. So we use the Kaja's model and work with regime. We publish this, we have this air-sea carbon dioxide flux. We have spring, summer, fall, and winter. Everything looks good. So like, for example, we can build a very simplified carbon budget that the export from the loop current can be largely balanced by the carbon dioxide influx from the air and the river input. So, but when I moved to Louisiana, things began to change. I take my first cruise with my family, use Carnival cruise. So I began to brainstorm by this kind of map from time to time. So what will happen in Louisiana is that if we don't do anything, 50 years from now, all these red pieces will be gone. And indeed, from 1930, the land loss in Louisiana is equal to the size of Delaware. And as we are speaking here, every 100 minutes, 1 football pitch of land will be lost. This happens 24/7. So you can see that maybe we will have no LSU Tigers like 100 years from now because we don't have enough football pitch, right? Yeah. So accompanied with this soil loss is a new problem is that we know that there is a lot of organic stuff in the soil. So if a quick calculation, we talk about the bay here, we call this a pointer or? This is a Bearterra bay here. So from this single bay that every year we're gonna lose about almost 2,000 gigagrams of carbon, which is about 7% of the carbon input from the Mississippi River. So this can be big. We need to take into the consideration about all this process. So to make this case even more complicated, if we take a close look at the Mississippi Delta, you can see that although we have overall land loss, we do have new land built up. For example, here shows this morning, but also by Tom's talk, we have this Wax Lake Delta building very fast from the land toward the ocean. Right? So then I take another cruise. So we go to the Bearterra Bay to see what's going on here. But you can see here, this is a newly emerged mangrove. We began to see more and more mangroves in Louisiana. So actually, we are experiencing this very fast transition from marsh to mangrove. And we know that in terms of carbon, there's going to be quite a different story between marsh and mangrove, right? But if we take a close look at this photo here, you can see, I will not see that after 2 years, there will be still a mangrove. For example, this is the root of the mangrove. It's no good. it's going to die pretty soon because of sea level rise and also because of the land loss. So to understand this, we got a project funded by NASA EPSCoR and together with Louisiana Board of Regents. We try to understand this, try to understand and quantify carbon export from the deltaic systems. And we will use these two sites. We will study from wetland all the way to coastal ocean, combining chemical oceanography, satellite oceanography, wetland ecology, numerical modeling, try to understand what is going on. Yeah. Thank you.
Great. Great. Okay, next we have Zi Feng from Hui, speaking about modeling the phytoplankton bloom dynamics on the Northwest Atlantic shelf, spatial heterogeneity, and the interannual variability.
Okay. Thank you, Margie. All right. So first, I would like to acknowledge my co-authors, Rubao Ji, Cabo Davis, Heidi Salsik, and Changshen Chen, and our funding agencies, NOAA and NSF LTR program. So in this work, we are adapting an intermediate complexity biogeochemical model, this 10-component model, to a high-resolution data simulative model for the U.S. Northeast Shelf, the FECOM, the finite volume community ocean model, the Gulf of Maine version 3. And this is also a model used for the Northeast Coastal Ocean forecast system. So at the first stage of this work, we are trying to understand how the model can represent the phytoplankton, the seasonality, and also the size composition in terms of small versus large phytoplankton. So I will show some results for this newly established LTR site from coastal to offshore, this transect, and also 2 representative stations in the Gulf of Maine and also Mid-Atlantic Bight. And so this site shows you the drastic seasonal differences in terms of phytoplankton biomass and size composition. On the left-hand side panels are LTR observations occurred in winter, January of 2018 and July of 2018. And the right panels are model climatology, 35-year model climatology for winter and summer seasons. What we can see is that our model did pretty good job in terms of how the phytoplankton biomass shift from winter to summer. We also represent this decreasing biomass from coast to offshore in the wintertime, pretty flat pattern in the summertime. And also in the wintertime, it's the large group dominated, and the summertime is the small cells dominated. And this is 35-year model climatology for the 2 stations I mentioned, the Gulf of Maine and Mid-Atlantic Bight. The point I wanted to make here is that the vertical physical structure makes dramatic differences for the phytoplankton productivity and nutrient regimes. So here we have the Gulf of Maine side, which are colder and fresher, and the Mid-Atlantic Bight warmer and saltier. You can see that makes difference in terms of how the nutrient replenished and depleted, and that sets stage for the chlorophyll distribution and size fraction. And we are working on relating the model phytoplankton structures to the physical processes. So stay tuned. Thank you.
Great. Our next talk will be by Christina Schultz from UVA, modeling the biogeochemistry and carbon cycle of the West Antarctic Peninsula.
Okay. So hi, everyone. I've been working on modeling the Antarctic Peninsula biogeochemistry. So I work with the Palmer Long-Term Ecological Research Program. And just to give a highlight of what we're looking into here, this is the region that I'm trying to simulate. And this is a region that has been changing a lot over the past few decades. So you have increasing temperature in the surface ocean, and you have decreasing sea ice. And that has led to several changes in how the ecosystem is working. So if you look at the northern part of the domain, you used to have areas that were covered by sea ice, and now they have less sea ice, which means that there's going to be more mixing and less phytoplankton. And if you look at the southern part, you have areas that were covered by sea ice, and then now their phytoplankton has access to sunlight, so it has increased, which is cool, but sea ice is not necessarily land-ocean continuum. However, if you look at the region and all the stuff that's happening there, you see that the freshwater budget is also depends on icebergs, on glacial melt, on precipitation that is influenced by land topography. And besides stratification and light, it's also, the freshwater budget also influences the amount of nutrients that's gonna come to this region, especially iron. So fast forward to some of the results from my model. What I'm showing here is dissolved organic carbon anomalies, and the reason I'm showing dissolved organic carbon is because it depends on both the physical part of the freshwater budget, so dilution and mixing, and also it reflects how much NCP is influencing the carbon cycle. So what you're looking at here is model data on the left, cruise data on the right, land close to the coast on the right here, open ocean, and these are all the different cruises that happened in the WAP, and the anomaly from the mean of each station. So basically, blue means less DIC that year, red means more DIC that year, and you can see that the models generally doing a good job at simulating the observed DIC, but sometimes you have something that goes not quite as expected. So here we're looking at 2011, in which the model predicts much higher DIC than what was actually observed. And this is an extremely low sea ice year that also showed this one of the only years for which we have dissolved iron, and you had incredibly high iron that's connected to glacial input. So this first highlights some of the obvious things we need to do in order to better understand the region and better be able to simulate it, which is to better understand the interannual variability of glacial inputs and what would be a good dissolved iron concentration that would reflect this glacial input. But it also highlights the importance of biology, given that this was a really large bloom, in driving DIC down and making this region be a potentially important sink of carbon, especially now that we have so much new CO2 flux data coming for the Southern Ocean. And I think the question of how big of a sink or source the Southern Ocean is still open. So I hope I made it clear that this is important to study.
Great, we have two more talks, and these are going to both kind of be related to the Arctic, which is going to segue into the Arctic mini session that comes next. So here we have Nikko Mayo from Bigelow, speaking on springtime coupling between Arctic sea ice shapes, the phytoplankton.
Oh yeah, it's okay, I will say it. So hi everyone, my name is Nikko Mayo. I'm a postdoctoral researcher at Bigelow. I'm working with Patty Matrai, and so today I'm just going to make a small introduction of a paper that we submitted a month ago, and we're talking about how the export of sea ice is influencing the biological production in the Greenland Sea. So first, I will just present the area of interest. So for this, I use some true color satellite images from the WorldView NASA applications. And so here you have Greenland. The Arctic Ocean will be here. The Greenland Sea is here. Svalbard is there. US will be here. Europe here. And so here you have Fram Straits, and the white part here is the ice. And I just take two images, so one in May 2011 and one in April 2016, almost at the same period. And you can see that you have like a strong interanual variability in the ice characteristics. So about like how you have this small scale variability here, but you don't have the same one here. So the question is about how the sea ice export will influence the biological production in the Greenland Sea here. Because one thing that we know is that the sea ice export from the Arctic Ocean is going to decrease in the next century. It will be no more sea ice at one point. So how the biology is going to respond to this? And so I try to use interanual variability observation of internal variability to try to have some ideas about what could happen. And so first thing is I look at how the internal variability in sea ice export is changing. So here you have the ice export during the month of April over 16 years. And so you have years with like higher sea ice export and years with lower sea ice export. And these two panels here is the sea ice concentration anomaly in the Greenland Sea. And so, of course, when you have more sea ice export, you have more sea ice in the Greenland Sea. And when you have lower sea ice, less sea ice export, you have less sea ice in the Greenland Sea. And so this is going to influence the physical certification, the biological production. I don't have time to present everything because I have only 3 minutes. But if you want to talk about this, just feel free to contact me. Thank you.
Okay, and the last speaker in our session is Ryan Woosley from MIT, speaking on freshening of the Western Arctic negates anthropogenic carbon uptake potential.
So hi, I'm more interested in the more larger scale basin uptake of anthropogenic carbon by the ocean. And the Arctic is thought to be a large sink for CO2 relative to its size. And the reason is the water is very cold, so CO2 is more soluble. And going into the future, as ice melts, you expose more surface area to the atmosphere, so you can get more air-sea gas exchange. So the question is that true? So there was a geotraces cruise in the Arctic in 2015, and this is the cruise track, and the colors show the PCO2. The PCO2 is very low relative to the atmosphere in the entire Arctic section. It's always lower than the atmospheric value. These are repeats of two cruises from 2005 and 1994. And so what I was looking at was decadal scale changes in these sections. And skipping straight to the results, the Western Arctic was very low in terms of anthropogenic uptake. So on the left, the Canada and the Makarov basins were much lower than the Eastern Arctic, the Nansen, Edmondson, and Eurasian basins, which were done by the German Arctic geotraces. And this is also low relative to the global average. Global average is about 0.5, so the purple line on there. So the Canada basin was about 5 times lower than the global average. And why would this be? Any carbonate chemist will tell you it's buffering capacity. That figure didn't come out very well, but there are other changes going on in the Arctic as well. So I was curious if other changes such as freshwater were influencing the carbon system. So I determined the water fractions, the amount of fresh water in the basin, so river water and sea ice melt, and looked at changes over the section. So on the left, you have the Canada basin. So in blue is 2015, and red is 2005. And so there was a large increase in river input over these 10 years. And what that means is that rivers have about half the alkalinity as seawater. And so when these mix, you lower the alkalinity and you lower the buffering capacity of the water. And so you also have to remember that there are 4 measured carbon parameters and 2 degrees of freedom. So on the right, you have PCO2 on the X, TA on the Y, the color is DIC, and the contours are pH. And so you have to know 2 of those in order to constrain the system. So if the green dot is the alkalinity and PCO2 from 2005, if you just have gas exchange, you increase the PCO2 by about 20 microatmospheres. You get an increase in DIC and a decrease in pH. But if you also decrease the alkalinity due to this mixing of fresh water, you get a decrease of DIC while still increasing the PCO2. So the important takeaway is to always fully constrain the carbon system.